Welcome to my channel. Today I am going to introduce you a very bright scholar of Indian Agricultural Research Institute. This is the video for those who are preparing themselves for ICAR JRF. Yes, we are going to meet Mr. Jayant, and Mr. Jayant has done his graduation from US Harvard. He has been the gold medalist of that university and he has secured All India 6th rank in Plant Sciences group. Thousands of students prepare for plant sciences and we are very lucky that we have Mr. Jayant here who have secured All India 6th rank in that examination. So today it's a golden opportunity to get insight from Mr. Jayant how to prepare your strategy, how to prepare for plant sciences. So Mr. Jayant, welcome to my channel, and our listeners are very excited to listen to you how you prepared for plant sciences. My first question to you is that the plant sciences is treated as one of the major group in ICAR JRF examination. So tell us more about the plant sciences group.
Thank you so much, sir. Thank you for a warm welcome. So first of all, coming to the plant science JRF or the agriculture as it is, I would like to congratulate all of you people for selecting agriculture as your career. As you know that agriculture has a lot of scope. Maybe in the future it was in the past and it will have it in the future. But coming to the plant science, that is the plant science scope. So plant science can be considered as one of the major subject or the major base for the agriculture, because as you all know that plant science includes the major subjects such as the genetics, the plant breeding, it may be the plant pathology, which is a very important aspect of the crop protection, and the microbiology, and it also has seed science and technology and the plant genetic resources. So these are the main 5 subjects which are included in the plant science, and you have to prepare all these 5 subjects if you want to get good marks in the JRF examination. And as you all know that this JRF examination is the national level examination which allows you to enter into the agriculture universities across the India. And one of the main benefit of qualifying through the JRF examination is that you are going to get a fellowship of rupees 12,640 rupees per month. So this is one of the main advantage of getting admission into the agriculture universities through the JRF examination.
Audience, you have to note one point that if you want to prepare for ICAR JRF in these 5 subjects, so you have to qualify the entrance in plant sciences group. So it is not the subject only it will come, but all the 5 subjects will come in the examination. So you have to prepare all the 5 subjects. So Jayant, please tell us about the books which you have referred during your preparation.
Yes, sir. If you people are interested in any of the 5 subjects, it may be the genetics, it may be the plant breeding, actually genetics and plant breeding will be considered in the one, and it may be the plant pathology, it may be the microbiology or seed science and technology. So if you want to get into any of these subjects, or if you want to do your masters in any of these subjects, you need to read, study all the 5 subjects. You need to start preparing for all the 5 subjects. So now I am going to mainly refer those important books for all the subjects and also for the general agriculture, as general agriculture makes a very important part of this examination. So first, coming to the genetics and plant breeding, which is considered as the major subjects within the plant science. So genetics, one can refer BD Singh, and Genetics Made Easy is a very important book which is like a gist of the BD Singh book. So you can refer the BD Singh book, and after reading the BD Singh book you can go for Genetics Made Easy. And for objectives you can refer BK Prasad and BD Singh. Objective Genetics by BK Prasad and BD Singh. So these are the main 3 important books that you can refer. You can also refer Kundan Singh, but if you read BD Singh it will be much better. According to me it will be much better than to read from Kundan Singh for the basic genetics, for basic understanding of the concepts. Now coming to plant breeding. Plant breeding, there are a lot of confusion. Students are in a lot of confusion whether to go for Kundan Singh or to BD Singh. So I would say that Kundan Singh is better because it has a very simple language. The concepts have been termed in simple languages. It is easy to understand. But for examination point of view, if you consider the examination as a main thing, so I would suggest BD Singh. For going for BD Singh would be better. And reading the BD Singh has one more advantage, is that if you come for MSc, when you are studying the MSc, even that would be helpful for MSc also. So I would prefer more of the BD Singh, or if you find it a bit difficult to understand, you can also follow Kundan Singh. And for objectives, you can prefer Kundan Singh objectives, and or you can also prefer this BK Prasad and BD Singh objectives for plant breeding also. But in BK Prasad and BD Singh, you have to be a little bit selective. Prefer only those questions or prefer only those topics which you are familiar with, or which you have read from your UG, or which the teacher has been taught. So prefer only those concepts, or else it will be a little bit hectic to read, and so that's the thing.
So Jayant, one thing I want to clarify here,
Yeah.
like there are certain books in all groups.
Yes.
Like we have to read it, like we have to read each and every page of those books. Like you told BD Singh and Kundan Singh.
Yes.
So up to what extent the student has to read these books?
Yes,
Each and
yes.
every line is important.
Yes, sir. I'm going to come for that.
Yeah.
So, as you asked from BD Singh, so this is the BD Singh book which I have. This is the Principle of Genetics, or Genetics by BD Singh. This is considered as the Bible of the genetics actually. So, from genetics, if we want to consider, I'll go little bit chapter wise. I'll go little bit deeply.
That would be very good.
Yes. So, if we come to BD Singh, yes, most of it are important, but you need to be selective. Like history of genetics, if you consider the first chapter, history of genetics will be most of the scientists, name of the scientists, how the things were discovered, how the cell was discovered, okay, how the genes were discovered, how chromosomes were discovered. So you need to cover a little bit of the basics, how they were discovered, who discovered them, what was the main basic or the principle behind that. So you need to cover a little bit of that, because you can see one or two questions based on these, the name of the scientist, and in which year they discovered that. So one more important is the organism that they used. Okay, the organism or the model organisms, so from which they discovered these things. So those are very important topics which you need to cover from the history of genetics chapter. Then coming to cell ultrastructure. Cell ultrastructure is like a very important topic for the examination. If you can read this cell chapter from 3 or 4 or 5 books, as much as sources you can, just read the cell chapter from everything, because it may be the cell wall. They will ask very key questions from this cell chapter. It may be the diameter of the cell wall, it may be the components of the cell wall, the primary cell wall, secondary cell wall, the tertiary cell wall, so and the nucleus, the cell organelles, what are the functions of the cell organelles. So these are the important topics that you need to cover, and you should also know which are the membrane bound organelles, which are the membraneless organelles, and whether they have dual membrane, whether they have a single membrane, and what are the main functions. Okay, and also we have mitochondria and chloroplast plays a very important role in this cell chapter, as well as for the nucleus. If you can, if possible, read the nucleus completely from whatever the sources you get it. If you read it from crop physiology book also, you can get very much brief knowledge about this cell chapter. So if you can refer different sources for the cell as well as for the nucleus, it will be much better. Then coming to chromosomes, yes, if you are preparing for genetics and plant breeding, it is very important that you should know about the chromosomes. So chromosomes, what is the structure of the chromosomes, who discovered chromosomes, what are the different types of the chromosomes, how the chromosome is organized. It may be the chromosome structure. There are different models of chromosome, like it may be nucleosome solenoid model. Okay, so and who gave that model in what year? So these forms the major questions that could be asked in your JRF examinations. And yes, you have different like karyotype, idiotype, the definition of those things, and what is heterochromatin, what is euchromatin, why it is heterochromatin, and what are the main regions of the chromosome that contain heterochromatin. Suppose we have centromere and telomere, which will be permanently in heterochromatin, and there are some like X bodies, the sex bodies, they will be like facultative heterochromatin. They are called as facultative heterochromatin because they will be normal euchromatin, but they will turn into heterochromatin. So they are also called as facultative heterochromatin. So you need to concentrate more of these technical terms when you are preparing for examination. Then coming to cell division, as you all know, cell division is a very important part. You need to read line to line in the cell division, because cell division, they might form a question from any part of the chapter, because cell division is such an important chapter that I prefer that you should read line to line in this chapter. And regulation of cell cycle, coming to regulation of cell cycle, I find it that it is not of much importance for this examination. I am again referring that it is not important for this examination. For the JRF point of view, you can skip this chapter as of me. So you can skip the regulation of cell cycle, but if you have time, please go through once, just through the surfaces. But again, in the regulation of cell cycle, you can mainly prefer what are the main checkpoints. You will find different checkpoints for different cell cycle phases. So you can refer checkpoints, and you have certain proteins like CDK protein, cell dependent cyclins. So those are the proteins which mainly regulate the cell cycle. So you can prefer those cell cycle, you can refer those cell cycles. So that would be more than enough from this chapter. Structural chromosomal aberrations. Yes, it is very much important to know about the structural chromosomal aberrations like deletions, duplications, transversions, transitions. So different types of those things. And major thing is what is the mitotic or the metaphase structure that will be formed due to it maybe deletion. For example, in deletion they form a loop-like structure. That is important. That might be directly asked. Those are the direct questions that you can find. And also the structure that will be formed in interphase nuclei. What are different structures that they form? And one more important thing is the type of gametes that they form. For example, in deletion they form n and n-1 gametes. So what are the type of gametes each structural chromosome aberrations gives us? That is one important aspect that you need to cover from structural aberrations, and you need to go through this chapter, very important chapter. And numerical chromosome aberrations, it is almost similar, that whatever you prefer in structural chromosome aberrations, similar things will be coming under numerical chromosome aberrations. Please go through this chapter very deeply, and very line to line reading has to be there for these two chapters. Then probability and chi-square test. Probability and chi-square test is little bit of a mathematics, I can say. It forms the basics for major of analysis. So the main thing that they asked from this chapter is that where we use the chi-square test. That is the one major thing that they use. So it is the only main question that is repeatedly appearing in the examination, where we are going to use the chi-square test. What is the main formula? There is a specific the history of the Mendel, the life cycle of the Mendel, what is the crop that he used, what are the 7 characters, and if you see there are the whole 7 protein characters, they are present on the 4 chromosomes, that is called as 4 linkage groups. So you should know which are the chromosomes that genes the Mendel studied were present. So those are the key points that you should refer from these two chapters. And also if you come to physical basis of heredity, that is chapter number 11, this mainly forms the chromosomal theory of inheritance, it may be, and the sex chromosomes, the white eye gene of Drosophila. These are some of the basic discoveries that were made. These were the milestone discoveries that were made in the genetics concepts. So you should know what are the main discoveries, what are the principle behind it, and who are the scientists that are involved and when it was done. And main thing is, whenever you are referring any discoveries or any scientist, please also refer what is the organism that they worked on. So this will be a very important aspect. You can see many questions regarding the Drosophila, it may be regarding the nematodes they worked on, regarding Arabidopsis, some work on Arabidopsis, some work on Drosophila. So these are the very important topics that you should also keep in mind while you study the discoveries or any milestones in the genetics or plant breeding concepts. Then we have gene symbols, dominance and lethal gene action. Yes, lethality or the gene dominance, it may be the dominance lethals, the recessive lethals, we have conditional lethals. There are different types of lethal actions for the genes, lethal genes. So you should know the definitions. Mainly important is the definition, and what are the conditions or what are the progenies that we get through this. It may be if there is a dominance lethal, what is the ratio in the F2? If there is a recessive lethal, what is the ratio in the F2? So you should know the ratios in the F2, and what are the organisms containing whether the dominant gene, whether they are dying, or the organisms having the recessive gene, they are dying. So these are the main things that you should prefer from this chapter. Then gene interaction, I would say it is a very, very, very important topic for the JRF preparation, because every year you see minimum 2 or 1 questions from this chapter. Gene interaction is a highly important chapter. You should know the definition of the all 6 gene interactions, 6 or 7 gene interactions. You should know the ratio, the F2 ratio, as well as the test-loss ratio. For each gene interaction, there is a different test-loss ratio. So you should remember these 3 aspects from this chapter. If you can remember these 3 aspects, that will be more than enough from the gene interaction. Then inheritance of a quantitative characters. Again, from quantitative characters, what are quantitative characters, what are qualitative characters, what is polygenic, what is monogenic. This will be important. And the major thing is the multiple factor hypothesis, which is given by Euler, which is a well-known hypothesis that you should know. What is multiple factor hypothesis, who gave it, on what crop. So that would form a very important aspect for this, as well as little bit of the effect of environment, whether the environment affect on crop or not, what aspect decided, and whether it is a monogenic will be affected by environment or polygenic will be affected by the environment. So these are the things that you should prefer from this chapter. But component of genetic variance again forms a very much key role in this chapter, that is whether it is an additive variance, whether it is governed by dominance variance, or whether it is governed by the epistatic variance. So based on this, the plant breeding will be decided. Whether if it is governed by additive variance, we go for pure line selection. Whether if it is a dominance variance, we go for hybrid selection or hybrid breeding. So additive variance is very important because based on this variance, so the fate of the plant breeding or the crop production will be decided. So please concentrate on components of genetic variance also. And the effect of environment and phenotype development, this is actually a very easy subject. This is because it's a common, it's like a general topic. It's like a story that you can easily read. So this just read on the surface. So no need to go for too deeper, or even if you don't understand a few topics, no need to worry much, but just read. It is very interesting to read also. So you can just read the chapter 15, which is effect of environment and phenotype. Then linkage in higher organisms. Linkage in testosterone generation is very important. What is linkage and how you detect the linkage and different types of linkage, these plays a very important aspects from this chapter. And linkage map, yes, they may ask the definition of the linkage map and the genetic distance, maybe the centimorgan or it may be the physical base pairs. So what is the linkage map, whether it is measured in centimorgans or whether it is measured in base pairs. Usually physical map will be measured in base pairs and linkage map will be measured in centimorgans. So these are the aspects that you need to concentrate in this. And factors affecting recombination frequency, yes, that is very important chapter. And psychological basis of crossing over. There are nearly 3 models that have been given based on this for a psychological basis of crossing over. Please go through those all three models, who has given them and what are the main aspects. That there are around 4 to 5 points in each model. So please refer the differences. What are the differences, who gave what is the difference between each model. So please refer that, because even that could be asked in the examination. Recombination in fungi and bacteria, I would say please refer transformation, transduction, conjugation and sexduction. So these 3 subtopics will be important, and remaining all for this examination point of view, you can skip these things, that is from 17.1 to 17.7. So as of me, for the examination point of view, you can skip these chapters, but please read 17.8 unit. That will be very important for the examination. Then molecular mechanism of recombination. For molecular mechanism of recombination, usually we won't see much of a questions. Even if there is a question, it will be usually one question, which will be very rare. So I would suggest that please don't waste your time on this chapter, that is the chapter number 8. If you don't have time, please skip this chapter, or if you are interested to read, you can please prefer this, but I would suggest that you can skip this chapter. Then sex determination in animals. You need to know mainly the primary and secondary sex characters. What are the characters? Then 19.4, 19.5, you can skip those because those are a little bit of physiological aspects. So you can skip those concepts. But you can refer the 19.7 unit, that is chromosomal sex determination, whether it is homomorphic sex determination or heteromorphic sex determination, and you should know the examples. For example, in birds and reptiles there is different system. For humans, for Drosophila it is a different system. So which are the systems and the perfect example for that? You need to remember all the examples for each system. So those will be very important questions for examination. Then sex determination of bigenic balance is a very important topic. Then evolution of X and Y chromosomes you can skip, that is not much of importance. Then coming to sex determination in plants. Again in plants you can just read it through the surface. Even we don't see much of questions from this chapter. So main chapter will be sex determination in animals. You must remember all the examples for each system of sex determination. Sex linkage, yes, sex linkage is very important. So sex linkage, you must study who discovered the sex linkage, in what organism they discovered the sex linkage, and what are the effects of sex linkage, what is the ratio, how the ratio changes if there is a sex linkage. Suppose we are getting 1:2:1 F2 ratio or 3:1 F2 ratio. If there is a sex linkage, what will be the ratio? So how the ratio changes. So these things you need to refer from sex linkage. and non-distinction of X chromosome is important. And what are the characters of sex-linked traits? Like sex, there are mainly three categories, like sex-linked traits, sex-influenced traits, and sex-limited traits. So please refer the examples of each categories, because it is very common that students get confused between these two examples, between these three concepts. So please refer the examples for each of the classes, like sex-limited, sex-influenced, and sex-linked characters. Please refer the definition also, as well as the example also. Then we have multiple alleles. Yes, multiple alleles is important. You should know the definition of the multiple alleles. And as of you know that humans, we have ABO blood group, which is again governed by the multiple alleles. So please refer the pedigree. Like if a man, there will be questions like if a man with A blood group marries with a B blood group, whether there is a possibility of getting a child of O blood group. So these kind of questions could be asked in examination. So please go through the blood groups very precisely. Please, if possible, refer some questions based on the blood group also. So those questions could be expected in the examination. And cis-trans test will be very much important topic. Recombination test and cis-trans test is important, and intragenic complementation is also very important. So intragenic complementation, the possibility is that they would mainly ask which is the organism that they discovered and who discovered this intragenic complementation. Then you should also refer the fine structure of gene. Fine structure of gene will be important, like recon, muton, and cistron. So repeatedly these questions will be asked in the examination. We can see many questions based on what is recon, what is muton, and as you can see that muton is the smallest unit, that is it needs only one nucleotide, and recon needs only two nucleotides. Recombination always occurs, it can occur between two nucleotides. So these type of questions will be asked. So muton is a basic, or we can say the smallest unit within the gene. So these kind of questions you should precisely know. Then comes mutation. So in case of mutation, the main things that you should concentrate in case of mutation is what is mutations. There are different types of mutations, maybe dominant mutation, recessive mutations. So these different types of mutations are important. And the main thing is what are the chemical mutagenic agents. Like there are some chemical mutagenic agents, there are some physical mutagenic agents, and the main the action of those things. For example, there are base analogs. There are acridine dyes which causes different types of action. They cause mutation in different ways. So what are the ways through which these chemical mutagenic agents cause the mutation? So those things you should know. So those forms are very important things in case of mutation. Then the detection of mutation won't be much asked in the examination. So I would prefer that you can skip the detection of mutation unit, that is 23.13 unit. So that you can skip from this chapter. Then cytoplasmic inheritance. Cytoplasmic inheritance, they mainly ask what is the maternal effect, the difference between maternal effect and the maternal inheritance. This you should precisely know the examples also, as well as what is the main principle or what is the main concept between the maternal inheritance and the maternal effect. This will be very important for you. And cytoplasmic male sterility is very important, because cytoplasmic male sterility is mainly governed by the mitochondrial genome. So this comes under cytoplasmic inheritance. So you should know about the cytoplasmic inheritance also. So that will play an important aspects for the examination. So this much will be enough from the cytoplasmic inheritance chapter. So coming to chapter number 26, that is the evolution and origin of species. So even from this chapter, there are very few questions that have been appeared in the examination. So I would suggest that please go through this unit number 26.2, that is theories of evolution, like Lamarckism, Darwinism, and mutation theory and neo-Darwinism. So most of the questions that have been coming from this chapter will be from this topic. So please refer this unit, and remaining unit you can skip this unit, because most of the questions we won't see from this unit. So please refer 26.2 unit from the chapter number 26. Then human genetics. In human genetics also, majorly in JRF examination, we won't see much of questions from human genetics. So I would say that you can skip the human genetics chapter also. Genetics of cancer, it is also of not much importance for the examination point of view. So you can skip the genetics of cancer chapter. Then genetic material, yes, the chemical nature and the structure. Please go through this chapter very precisely, like line to line reading is required from this chapter, because this forms the basis for how the gene was discovered, how the DNA was discovered, and who discovered them. These are very important topics, and there are different experiments like Griffith experiment. We have Hershey and Martha Chase experiment. On what organism they did their experiment, and the year of experimentation. So these are very important aspects that you need to cover from this chapter. And the structure of DNA, yes, they will ask the primary structure of DNA, and the whole structure. It may be the phosphate backbone, it may be the sugars, what are the sugars that are present, what are the nucleotides that are present. We have ATGC in case of DNA and AUGC in case of RNA. So these kinds you need to concentrate more from this chapter. And one more important thing is you should know the different characters of different forms of DNA, that is A form, B form and Z form. There are majorly 3 forms of DNA. Please refer all the characters of these 3 DNA types, because any one could be asked. Just it may be the whatever the diameter. There are different diameters for different types of DNA. It may be the length, the pitch of the DNA is different for different types of DNAs. And what is the in which condition A DNA is predominant, in which condition B DNA is predominant. So these kind of questions will be asked in the examination. So please go through these differences or these characters of each types of DNA. Then genetic material, the organization, that I said as like how the DNA has been organized. It may be nucleosome solenoid model, because we have different types of models we have. So please go through that models. And C-value paradox forms a very important aspects in this. But you can skip this genetic material part 2, that is organization. With respect to 30.4 unit, it may be 30.5 unit. And organization of genes will be little bit important. You need to refer that is the 30.8 unit, as well as 30.7 unit, that is unique and repetitive DNA. They will ask what is unique DNA, what is repetitive DNA. That would be a little bit important topic. So except to this, you can skip other units from this chapter for the exam point of view. Then genetic material, that is the multi-gene families. So from this chapter you can expect some questions like what is multi-gene family, and what is they have repetitive genes we have. So those kind of questions you can expect, like what is pseudogenes we have, how the gene family origins. We have different examples for gene family. For example, if you take myoglobin genes, or if you take like those kind of genes, they have originated from multi-genes. They are called as multi-gene families. So how they originate? It may be mainly the multi-gene families, they originate through mutation and recombination. So duplication and mutation. So these are the things that you need to concentrate more from this chapter. And you can skip the unit of 31.6. So that would not form a major aspect of this chapter, as well as 31.7 unit, you can skip that. But please refer the gene clusters and gene families. You should know what is a gene cluster and what is a gene family. The major difference between a gene cluster and a gene family you must know. Then genetic material, that is the replication. So if you come for replication, yes, the whole process of replication is important, very much important. And what is the main principle, like whether it is a semi-conservative, whether it is a conservative, whether it is a completely dispersive replication. There is 3 experiments that have proved that the DNA replication is semi-conservative. Please go through the experiments, each experiment, and who has conducted those experiments. Please go through that. And coming to replication, there are the major things is what are the major enzymes that are involved. Okay, there are helicases, there are single strand binding proteins, there are DNA polymerase 1, 2, 3. We have different types of DNA polymerases, and there are different activities of each polymerase. If you take DNA polymerase 1, there are 2-3 activities for that. If you take DNA polymerase 3, we have like 5' to 3' exonuclease activity, 5' to 3' polymerase activity. So please go through each activity of each polymerase enzyme. So if you find it difficult to read it from this book, so please refer a Genetics Made Easy book. In Genetics Made Easy book, they have given this chapter very precisely, very briefly, and you find it very easy to refer from that book for this concept. That is the enzymes for replication. What are the enzymes? What are different enzymes, and what are the components, what are the functions of each enzyme. You can refer a Genetics Made Easy book. And a replication of 3' end, that is the telomere replication, also very important, which is the enzyme that carry out the telomere replication. That would be a very important aspect. Then DNA repair. Again we won't find much of questions from the DNA repair. So if you can read, please read the mismatch repair, the excision repair, yeah, these two will be these two will be enough from the repair. And DNA modification, if you can read, please read DNA modification. So apart from this, you can skip this. You can skip this chapter also. Then molecular techniques in genetics. So this you can read the basic concepts, whichever the concepts that you have been taught from your UG. So you can refer only those concepts, because whatever the concepts that have been given in this, they are little bit of molecular types or biotechnological types. So this would be little bit advanced for for this examination. So you can refer the hybridization. You might have all read through your biotechnology course what is southern hybridization, what is northern hybridization. So mainly concentrate in what aspects they are used. For example, if you use southern hybridization, you are mainly going for DNA-DNA interaction. If you are using northern hybridization, so you are mainly going for the DNA-RNA interaction. If you are using western blotting, you are going for DNA-protein interaction or protein-protein interaction. So these kind of topics, if you can remember, that would be enough. So no need to go for too deeper from these chapters. Like there are fish, there are techniques like fish, there are like GISH, that is genome in situ hybridization, the fluorescence in situ hybridization. So if you can remember the major uses or utilizations of those techniques, that would be much more than enough for the examination point of view. So please don't go too much deeper if you are finding it difficult to read. So no need to go too deeper. So please go if you can remember the uses and what is the main principle, how they work, how each technique is working. So if you can remember these things, that would be more than enough from this chapter. And please read markers. Many students have very much problem with the markers, because markers is such a subject that few people won't understand, because you need a lot of imagination to understand the markers. So that is one major issue that the students find these days. So through the markers, I would suggest that if you can remember whether that marker is a dominant marker or a codominant marker, and whether it is a multi-allelic or whether it is a biallelic. So if you can remember these things, that would be more than enough from the markers chapter. And genome maps. Genome maps, again you will get all the definitions of genome map. What is a linkage map, what is a cytogenetic map, what is a physical map. You will get the definition. So that definition will be enough for this. And genetic markers, as I said, whatever the topics, whatever the concept that I said from the genetic markers, that much you can remember. That will be enough for you. And from cloning, that is the unit 35.6. That is AFLP we have, sequence tag we have, map based cloning we have. So these are little bit of advanced level for this examination, I would say. So you can refer what are the main principle behind those things. And if you find it a bit difficult, you can skip also because we will find only one or two questions which related to marker. So please go through whichever the aspects that I said from the marker point of view. Then chapter number 36. So we wouldn't find much of questions from chapter number 36. So you can skip this chapter number 36 because it is much of a biochemical point of view, which are the chemicals that are affecting the human, what are the diseases that different chemicals causing human diseases. So this you can skip with respect to the examination. So 36 chapter you can skip. And coming to gene action, that is the transcription. Again, how you read the replication, please read the transcription in the same manner. You refer mainly on what is the process, how the transcription has been discovered initially, and what are the main enzymes or major enzymes that are being carrying it in transcription. And also in transcription you will have this RNA processing, that is splicing. So mainly in RNA processing they majorly ask about splicing. So apart from splicing, there are different kinds, like there are modifications we have different mRNA modifications, we have RNA editing. So those things need not to read for the examination. So if you can read about this splicing, so but you need to read completely about this splicing. Splicing is a very important aspect with respect to this RNA modification. So please go through the splicing unit from this 38th chapter. So that will be enough for examination. Then translation, again translation is you come across the genetic code, it may be what are the stop codons, what are the start codons, those you need to remember, and what is where the transcription occurs and where the translation occurs, because transcription occurs inside the nucleus and translation occurs outside the nucleus. So how the RNA is transferring from nucleus to the cytoplasm. So what are the different mechanisms that you can refer from this. And you can also refer what are the different types of amino acid tRNAs, which there is one more hypothesis that is called as Obel hypothesis. Obel hypothesis is very much important because it is the main thing that is giving for this protein diversity. By using this 64 codons, we are from only 20 amino acids, there are 64 codons. So how this 64 codons are coding for 20 amino acids. So this is very important aspect, that is the Obel hypothesis. Please refer Obel hypothesis also, and who gave this Obel hypothesis, and characters of genetic code. There is universality character, there is non-ambiguity character, and genetic code is a degenerate. So please refer these characters, because there will be very precise questions from this. Suppose they will give that right, true or false, whether the genetic code is non-degenerate. So if you don't remember or if you don't read precisely, you will not be able to answer in the question. So please read the characters of the genetic code very much precisely. And then protein transport and signal transduction. Again, from this chapter number 40, we won't find much of a questions. So I would suggest that you can skip this chapter. So from this chapter, that is regulation of gene action in eukaryotes. So you can go through this unit number 43.5, that is dosage compensation. We have dosage compensation mechanism in different organisms. In different organisms, there is different mechanism. For example, in humans, we see that in females, one of the X chromosome is this one, bar body. It is converted into bar body or heterochromatinized. So like this, we have different mechanisms in different organisms. Please go through the different mechanisms as well as remember the examples for each mechanism. There are majorly 3 mechanisms. So please remember the examples also. So this much will be enough from this chapter 43. So remaining all you can skip. Then genetics of differentiation and development. Even in this chapter, not much questions are asked in the examination. So this chapter also you can skip. So please go through whatever the chapters that I have said. But from genetics point of view, if you can consider genetics, it forms a basic of the plant science, because it is a major subject that most of the students prefer. Most of the people take plant science because they are interested in genetics. So if anyone has a very interest in the subject, please go through the book. But if you are preparing majorly for the examination point of view, please be selective, because if you read too much also, it is a problem. So remembering too much of information is again a problem for you. So please be selective. Please do little bit of work on what to read and what to remember. So this much will be, I think, helpful from this genetics point of view. And so that would be it from the genetics.
One point which I am getting is this book BD Singh, student has to read like Bible.
Yes, sir. It is actually a Bible.
Accepting some of the few chapters you suggested on the basis of your experience, that they can escape. But ultimately you should have this book, you should jot down the notes from this book, and जैसा हम कहते हैं ना कि we are doing breakfast, dinner, lunch with that book only. So you have to be thorough with this book. So this is the basic of this book, and I am very fortunate. When I was in BHU, I met Dr. BD Singh sir. He was rector at that time. So very humble person, and this book in graduation, when you are narrating all the important points, I reminded of my graduation, and you thoroughly revised the whole syllabus. So wonderfully explained by Jayan. You should jot down the notes, you should jot down the points which Jayan has told, and you should start from this book. So apart from this book, what are the books and what is the strategy from plant breeding point of view?
Yes sir. Coming to plant breeding is like a little bit of advanced or the application top kind of thing, because genetics is the basis for plant breeding. Again, if you know what is genes, if you know what is chromosomes, you can apply the plant breeding techniques. So this forms like an advanced topic, and plant breeding, yes, it forms very important. This is the plant breeding book from BD Singh. So this is the plant breeding, the principles and methods from BD Singh. As again I said that most of the students, they are confused between whether to go for BD Singh or whether to go for Kundan Singh. So from Kundan Singh, if you can read, it is little bit, I can say it is easy to understand. But for the examination point of view, if you want to cover much of a topics, if you want to cover more things, more information, please go through BD Singh also. If you have much time, you can go through BD Singh. But you might find it little bit difficult to understand from BD Singh. So please refer any professors or any teachers, so that you will have a better understanding of the topic. Because this is, as I said, this is a little bit of application level thing. So you should have understanding in this plant breeding. There is not much of mugging up, as I can say. There is not much to much of remembering from the plant breeding book. But you must have a very good understanding of each concept and each aspect from this book. So now let me just introduce to what are the major topics that you need to cover from this book. As you all see that introduction of plant breeding, again we will have the scientists, who are the scientists, how the plant breeding developed, what are the historical aspects of plant breeding. Please go through that, the scientist or the year, what are the plants, so what are the crops they worked on. Please go through the issues from introduction of plant breeding. Then domestication and germplasm conservation. Yes, they will ask like what is germplasm, what is domestication, different types of crops we will have. We will have land races, we will have we will have elite varieties, we will have these kind of things are important, definitions of these kind of things. So that will be important. The yes, absolute varieties, we have varieties in cultivation, breeding lines. So what are absolute varieties, what are land races? So these things you must read from this book, from this chapter, that is domestication and germplasm conservation. And center of origins. Yes, center of origins is very important because for each crop, then you can see in the question paper that they will suddenly give what is the center of origin for chickpea, what is the center of origin for rice, what is the center of origin for sugarcane. So please go through the general agriculture book. You will get this in general agriculture book. I'll refer which general agriculture book you should refer. So please go through general agriculture book. Please read which crop is originated from which country. So that you should remember. So that is a very important topic from this chapter, as well as collection of germplasm. Whether there are different methods for collection of germplasm. So how they collect the germplasm, how the germplasm is stored, because there are gene banks, there are seed banks, there are field gene banks. So what are these gene banks or field banks? So, and what are the major crops that are being stored? For example, if you have any exotic species or if you have any endangered species, so those will be mainly conserved in case of gene banks. And if you have any endangered, so those will be conserved mainly in the gene banks. They will extract the gene from any found varieties. They will conserve in the gene banks. If you have any crops where the seed will not be viable for much time, so they go for field gene banks. They will grow it in the field year after year. They will conserve it. And majority of the crops where the seed is the propagating material, they mainly go for seed gene bank. So please go through these gene banks, different types of gene banks. What are the advantages and disadvantages of each gene bank? Please go through those things. And center of origins, I said. And in situ and ex situ conservation, this will be a repeated question in examination. What is in situ conservation, what is ex situ conservation, and the examples where they do go for in situ conservation, what is ex situ conservation. So please go through examples as well as definition from this chapter. Then you can also refer IBPGR and NBPGR. So when the IBPGR was established, where the headquarters of IBPGR is, and where is NBPGR. NBPGR, as you all know, that it is in the near to IRA campus, that is just beside the IRA campus. So, and when it was established, what are the main functions of NBPGR? So please refer IBPGR as well as NBPGR. So these two institutions are very much important with respect to germplasm. Then plant introduction. And what is primary introduction, what is secondary introduction, and what are the different crops that have been introduced to India. So there are many crops that have been introduced from foreign to India. Please go through which are the different crops that have been introduced, and if you can remember, please remember during which time. So that will not be much important, but if you can remember, please remember those things also. And important achievements they might ask, and major questions that people ask is whether we need to remember the varieties or not. So varieties name, because there are lot much of varieties in the the mentioned in the book also, and there will be lot much of varieties that will be released. So with regard to varieties, I would say that, so please go through which are the recent varieties that have been developed, majorly from, usually it will be from IRI. So major varieties that have been developed and released through IRI. So those varieties will be majorly asked. And if you can go through some of the varieties that have been developed through mutations, mainly mutagenic varieties. So that will be important. And if there are any varieties from IHR Bangalore, those varieties will be little bit important. And also those varieties which have been developed through transgenic varieties, again they will ask what are different types of transgenic varieties we have, what are different transgenic, like BT cotton we have, and what are the main features why that transgenic crop has been developed. So that will be asked. And also the somoclonal varieties. We have somoclonal propagation in case of sugarcane. In sugarcane we can see a lot of clonal varieties. So please go through these varieties. These varieties play an important role. But you need not to remember all the varieties from all the crops, because that will be again a lot of information to remember a lot. And modes of reproduction in plants, again, which is a very important topic. Please read line to line from this chapter. That is the modes of reproduction. It may be self-propagation, so selfing, or it may be cross-pollination, self-pollination. It may be often cross-pollination. Even often we can see that there is, if the pollination percentage is from 5 to 30%, we call it as often cross-pollination. So what must be the pollination percentage to consider whether it is a self-pollinated or often cross-pollinated or cross-pollinated? And how you detect a plant is self-pollinated or how you detect a plant is cross-pollinated. So please go through these aspects from this chapter number 4, and please go through apomixis. Apomixis plays a very important role, and also read what is the genetic constitution. If you go for self-pollination, what will be the genetic constitution of the population? If you go for cross-pollination, what will be the genetic constitution? So genetic constitution also, they will form major questions from the genetic constitution. So that is very important. And self-incompatibility and apomixis. From self-incompatibility and apomixis, again, self-incompatibility is very important aspect in case of controlling the pollination in crops, because if there are some crops where hand pollination is very difficult, in such crops we use self-incompatibility. So as you are going to be a plant breeder, you must know how self-incompatibility works. There are different like gametophytic self-incompatibility, sporophytic self-incompatibility, and what are the alleles, or there are different genes that govern these things, and what are the effects, like when you cross the two different genetic constitutions, what the result you are getting, whether it will be incompatible, whether it is partially compatible. Those kind of aspects which in the book it has been clearly mentioned. There is a table for that, so please go through that table, and that will be a very important multiple choice question for the examination. And pollination control, again, male sterility and self-incompatibility, you must go through line to line, because these two are very much important pollination control mechanism that have been widely used for plant breeding aspects. So please go through line to line from these two chapters. Please go through like the different sources of male sterility. For example, if you take wheat, there are different sources. For rice, we have different sources. What are the different genes that are governing it? Maybe the male sterility, genetic male sterility, or cytoplasmic male sterility. In onion is mainly breeded by using cytoplasmic male sterility, that is CMS. So please go through these type of concepts from these two chapters. Please read line to line from these two chapters. Then qualitative and quantitative characters. This will be usually coming under genetics also, the basic genetics also, that is what is quantitative, what is qualitative. But here you can read all the definitions, like what is dominance, what is pleiotrophy, what are segregants, we have transcriptive segregants we will get. So please go through the definitions of each one. So definitions will be important from this chapter. And how the qualitative traits will be inherited, whether we have monogenic, whether we have polygenic, how we need to breed. That is, for polygenic traits we have different breeding methods, for monogenic traits we have different breeding methods. So please go through which are the different breeding methods we use in different crops, and that is important aspects in case of qualitative or quantitative genetics. Then biometrical techniques in plant breeding. Usually biometrical techniques, you will find one or two questions, but I would say that if you have not been taught in your UG, UG courses that about these biometrical techniques, it would be a little bit difficult for you to understand. So please refer any professor or any of your teachers. And so in summary, to refer from this again the plant breeding methods. So you prefer the applications. What are the major applications of each plant breeding methods? Like what are the population that we are getting? For example, from backcross population. From backcross method, what is the population? For example, from backcross we will get near inbred line, that is NIS will be obtained from backcross. From pure line, obviously you will get a pure line variety. So from each breeding methods, please refer what is the principle, what is the main use, and what is the outcome of each breeding method. So no need to go for the whole procedure, what we do in each year, what is the selection procedure. Please no need to go for each procedures of breeding methods. So please refer these 3 or 4 concepts which I said from breeding methods. And again, bulk method, SSD method, and also please go through who are the scientists that have been given each breeding method. Which scientist gave which breeding methods? Please go through that. That is also very important aspect. And we have cross-pollinated again. Population improvement. Population improvement again plays a very important aspect from this book. So please go through population improvement methods. Like it may be recurrent selection, it may be RSS for GCA, recurrent selection for SCA, reciprocal recurrent selection. Please go through the concept, and many students might find little bit of difficulty in understanding the this one population improvement methods. If you find any difficulty, please refer to your professors and go have a proper understanding of these concepts, because understanding is very much important, because we find a lot of application level questions from plant breeding aspects. So please understand the topic rather than just remembering everything. From the plant breeding, you need is understanding. What you need is a complete understanding of the concepts. So please go through this recurrent selection, RSSCA, RSGCA. So please remember what are the different types of, like if you RSGCA we use mainly the genetic combining ability. For SCA we use mainly the specific combining ability. So please go through these type of aspects from this chapter. Then again, hybrid varieties. how the hybrid will be developed, what is the inbred, how we go for inbred development first. First we must have an inbred to develop a hybrid. So first how the inbreds will be developed, what are the major characters that we need to consider before developing an inbred. So and how the hybrid will be developed, how the crossing will be done, in which crop we have, in each crop we have different types of crossing. For example, if you take sugarcane, we have different like Latin method we have different type. For example, if you take pulses, we have different types, like we have emasculation technique like Shiva Shankara method. So we need to concentrate more on special methods in each crop. In each crop we have a special method of hybrid development or the emasculation technique. So those kind of things you need to read from these chapters. And again we have cytoplasmic male-sterility. For seed production they will use CMS, they will use GMS. In which crop we are using GMS? In rice, as you can see, we use GMS, CMS as well as CGMS. In rice all the three types are used. So and in onion, as I said, onion and most of these crucifers we use CMS system will be used. So please go through these systems and what are the donors, the species, which species are donating this CMS system or CMS genes. So please go through these aspects from hybrid. And hybrid sterility we have, hybrid inviability we have. So these kind of aspects you need to remember the definition from each of this. And as well as synthetics and composites. So what is the population, whether it is homozygous, heterogeneous, we have four type of populations: homogeneous, homozygous, homogeneous, heterozygous, heterogeneous, homozygous. So these kind of populations and example of each population. For example, the pure line is a homozygous homogeneous population and a hybrid is a heterozygous homogeneous population. So you need to know the example of each population. So that will be again very much repeated questions that we can see in the JRF examination. Please go through top cross. What is top cross, what is back cross, what is test cross. So please go through these topics also. Then we have method for clonal crops. In clonal crops again they will ask clonal selection is mainly followed in vegetative propagated crops. So please go through what is clonal selection, how the clonal selection is done, and what are the major varieties that are being developed through clonal selection. So this will be important from clonal selection methods. And stress resistance, again coming to stress resistant breeding for stress resistance, it may be biotic stress or it may be abiotic stress. So if we consider these two chapters, the major thing is you need to read there are some chemicals. For example, if you take abiotic stress, in case of drought, the proline content of the plant will be increased. So you need to concentrate more of these kind of topics. Which amino acid is increasing, which carbon or which sugar is increasing, what is the any compound is metabolite is increasing, in under which stress, whether it is a cold stress, and heat stress we have HSP genes, that is heat shock protein genes. So these kind of topics you need to cover from stress. No need to read each line from this chapter because this is a very elaborately they have given these things. So please remember these kind of things. Usually they will be given in a table or some table will be given for these kind of things. So please remember this. And coming to the pest resistance, that is the biotic stress resistance. So in different crops we have different mechanisms. Different crops have developed different mechanisms to avoid the pest. For example, in case of pest we have pest invasion, we have this one, we have antibiosis, anti-zoonosis, we have non-preference. So what are these methods? Okay, you need to remember the definition of each methods. And for example, if I say in cotton, the hairiness or the pubescence on the leaves or on the stem, it repels some of the pests. So if you take the biotic stress resistance, as I said that you need to know what are the different mechanisms that each crop has adapted to. It may be the evasion of the pest, it may be avoiding, or the pest tolerance, it may be the antibiosis, anti-zoonosis. Please go through these kind of topics and please remember the examples or whichever the chemicals that the plants are producing or whichever the morphological characters that the plant are adapting. So please go through these type of examples and aspects with respect to the examination point of view. So that would be much enough from the breeding for biotic and abiotic stress resistance. And if there are any important varieties, for example, we have in wheat we have drought tolerant variety like C360, and we have in rice the sub-tolerant variety, that is Swarna sub 1. So these are some of the important varieties that are measured for JRF examination. So if you can remember those kind of varieties, those important varieties, so please remember the important varieties. So those will be given in the book itself. So you need to go selectively, you need to remember selectively. So please remember only the important varieties you need to remember. Then we have idiotype concepts in crop improvement. Idiotype, you need to remember the idiotype of major crops like rice, it may be the sugarcane, pulses, we have wheat, and the cotton we have. So some of the major crops idiotype you need to remember. There will be some around 10 to 15 points for each crop. That is what is idiotype, what are the different characters that the plant should have to call it as an idiotype. So please remember the idiotype of some important crops. So that would be more than enough. And how idiotype breeding will be done. So please remember those. They have given some of the selected crops, the idiotype of selected crops. So please go through the idiotype of selected crops. And breeding for quality, that is the protein quality, it may be the amino acid, increasing carbohydrate or increasing the vitamins. So in this case, you can go for like, for example, if you take maize, we have the HQPM, that is quality protein maize. So these kind of varieties will be important. That are what are the different genes that they have incorporated into the plants. For example, golden rice. Golden rice is enriched for vitamin A. So what are the different genes for golden rice? What are the genes that have been incorporated into the rice? So from which source they have incorporated? What are the sources, different sources that have used, and when it was developed? Who was the father of golden rice? Who is father of super rice? So please go through these kind of aspects. So they will be repeatedly asked in the examination. We can see these questions, and mainly the protein rich varieties, different varieties. It may be maize, it may be sorghum, it may be rice. So these questions will be repeatedly asked in the examination. So please go through these aspects. And breeding for oil quality, yes, in oil quality you can see that the cotton, the mustard, maybe if you can see mustard, and most of the crucifers or oil crops. So and oil palm, they will ask most of oil palm. Please go through the what is the different oil content. Okay, the oil content of different crops. Majorly we see the oil content of different crops. Usually in cotton seed, how much oil content will be there. So and even rice bran oil, rice oil or rice bran oil. So these kind of questions will be repeatedly asking from this chapter. So if you can go through that, that would be more than enough for oil quality. So you need not to go for different breeding methods that have been used in oil quality, but if you can remember the major varieties or major content, whether it is amino acid content they are increasing, so what content they are increasing. So if you can remember those things from this chapter, that would be more than enough. Then mutation in crop improvement, again you will be reading the mutation, the same chapter in the genetics from BD Singh book also. So if you have read that thoroughly, here you need not to read again the what are the hybridizing agents. It may be mutagenic agents, it may be physical mutagens or chemical mutagens. But what you need to remember from this chapter is how the mutation breeding they carry on, how mutation breeding is carried out. For example, in some they use like gamma garden will be used. So how they will be carrying out the mutation breeding, and what are the important varieties that have been developed through the mutation breeding. So these concepts you need to cover from this, or you can skip the mutation chapter from the genetics book and you can read thoroughly from the BD Singh of plant breeding also. But go through this chapter very thoroughly, read line to line, because mutation chapter is such a subject, such a chapter that from every year we could see at least 1, 2 or 3 questions from the mutation breeding. So please go through mutation very deeply. And polyploid in plant breeding, again they will ask what are polyploids, different chromosome numbers of polyploid crops, because we have a lot of polyploid crops like sugarcane. We have most of we have Nagarro's U triangle for polyploid crops. So please go through the chromosome number of each crop, because chromosome number again it is very much important. And if you come to sugarcane we have sugarcane breeding, we have special breeding techniques in sugarcane. So please go through the special breeding techniques of sugarcane. We have cotton and we have tobacco. So these are very much important, like commercial polyploidy crops that have been cultivated. So please go through these crops for polyploidy plant breeding. Then what are alloploids, what are aneuploids, monoploids, again whatever you have read in structural and chromosomal numerical aberrations. So similar thing you need to refer from this aneuploids, monoploids, triploids, tetraploids similarly. Like what are the different structures that are forming in case of interphase nuclei, what are different structures that are forming metaphase nuclei, what are the fate of the gametes, how the gametes will be formed, whether it is n gametes, n+1 gametes, n+2 gametes. So when you cross monoploid or monoploid with diploid or aneuploid with triploid, so what are the progenies that you are getting, how the genetic constitution of the progeny will be. So that you need to thoroughly know from this chapter of polyploidy in plant breeding. Then distant hybridization. In distant hybridization, what you need to cover is how the crops have been originated. For example, wheat, There are three different types of wheat. Mainly the diploid, we have tetraploid, and we have hexaploid. How the hexaploid wheat have been originated. And for example, in case of rice, we have mainly two: Oryza sativa and Oryza glaberrima. How these they have been originated. And in case of cotton, we have Gossypium. How this Gossypium species has been originated, through which crosses, from which wild species, what is the progenitor. So these kind of things you need to remember from this distant hybridization. Mainly this will be asked from the distant hybridization, that is the origin of important commercial crops. Okay, tobacco it may be, the cotton it may be, Nagarus U triangle you will get. So please remember these things. And also remember the barriers in production of plant hybrids, because when you cross wheat and maize, or there are some barriers. For example, if you have indica into japonica, there is some barriers. If you cross wheat, different types of different ploidy wheats, there is some reproductive barriers. What are post-reproductive barriers? What are pre-reproductive barriers? Please go through these kind of reproductive barriers, and please know the little bit of examples. Examples is not much of importance, but if you can remember, please remember the examples also. Then biotechnology in crop improvement. Biotechnology, as you all know, that you would be studying a separate course for the biotechnology in your UG studies. So if you have a very good notes of your UG studies, you can refer that notes, or if you don't have sufficient notes, please refer this chapter from the BDSIM itself. So this is very clearly given. You can just refer, you can go through a little bit of surface, no need to go too much deeper in this, but please remember what is the nutrient media, what are the different chemicals that they use, how tissue culture is done. Okay, please remember little bit of surface, that much will be enough for the biotechnology. Again, you will have embryo culture, when the embryo culture will be done. Okay, when tissue culture is done, what are different steps for the tissue culture, and major varieties if they have developed through tissue culture. For example, banana is mainly propagated through tissue culture nowadays. So please remember those kind of things in case of biotechnology. And one more aspect you need to remember is about the agrobacterium mediated transformation. So that is very important thing, because you need to remember what are the different genes. There are different genes that are regulating in agrobacterium mediated transformation. So please go through the agrobacterium mediated gene transfer also. And somatic cell hybridization, yes, somatic cell hybridization they will ask, and they will ask how the protoplast is fused, protoplast fusion. For example, we have electric fusion, we have by using chemicals, we give some chemicals so that the cell will become competent and they will fuse easily. So these kind of things, again we can see repeated questions from this kind of aspects. So please go through thoroughly for these concepts. And again, molecular markers, as I already said, please go through whether it is dominant marker, recessive marker, or it is allelic. What is the allelic relationship? So this much, if you can remember, that will be more than enough for molecular markers. And for varieties, again as I said, if you can remember major varieties, that would be more than enough. No need to mug up all the varieties that you come across. And quality seed, this chapter number 38, if you see that you are going to read almost similar topics from the seed technology also. So if you don't have time, I would suggest that you can skip this chapter, or if you find interesting, or if you need to read, please go through this chapter also. This is also very easy chapter, but you need to remember things from here. From this chapter you must remember most of these things, because seed technology will be merging with this chapter. So you can read it through seed technology or from this book also. Then organization for crop improvement. In organizations, usually not much questions will be asked. They only ask which crop, the ASERP of which crop is where. Where is this? For example, Indian Institute of Farming Systems, it is in Modipuram. So like that. For mushroom research, it is in Solan. So these kind of questions will be asked from organizations. So you can skip this chapter. So just go to ICR website. In ICR website, you will get the whole list of NRCs. It may be the ICR or the different research institutes. You will get the whole list. So please go through the whole list of it, and please try to revise as much as possible so that you can remember all the different research institutes and their places. And international institutes, again international institutes, with regard to international institutes, you need to remember what international institute is working on which crop. For example, we have CIMMYT in Mexico, which is mainly working on maize and wheat. Okay, so these kind of things you need to remember from this, and when it was established. So if you can remember these things, so that would be more than enough. No need to go for like what are the functions of these institutes and why it was formed. So no need to go for too much deeper from this. So which crop it is working on and when it was established. So that will be much enough for institutes, with respect to central institutes or international institutes. Then intellectual property rights. In intellectual property rights, they mainly ask what is PPVFRA. Okay, we have PPVFRA, we have, and what is the act, when the act was enacted, and what is the main regulations under the act, what is the different principles that they have following, and what are the benefits that the farmers are getting through this act. So these kind of things they have been given in the book. So please go through these kind of things. You can see maybe one or two questions from this chapter also. So please go through farmers' right. It may be breeders' right. There are several benefits that are given only to the breeders. There are several benefits which are given only to the farmers. So please go through these aspects from the intellectual property rights. But to summarize that, plant breeding is mainly how you understand the things. It is not that as genetics you need to remember much, but plant breeding is how you understand, how you interpret, and how you finally you give up your output. So in examination also you can see that there are much of application level questions from the plant breeding. So please try to understand the concept as much as possible. So if you are having any doubts, please refer someone, refer any professor or refer any subject matter specialist. Please try to understand the concept from the plant breeding. So that's for the plant.
Jayant, this is for plant breeding, and so we can say that genetics will build up your concepts,
yes.
Your basic concepts, and the application part of that concept will be cleared by the plant breeding book by BDC. So one more thing I want to ask you, Jayant, is there are other subjects also in that
yes,
group, like plant pathology.
So H.C. Dubey is one of the major books that most of the students refer for plant pathology. So but in H.C. Dubey you need to cover again the history will be there. What is the history, how the plant pathology was being discovered, what are the different diseases that have been discovered through the time, through the history, and who discovered them, which is the first bacteriological disease, which is the first fungal disease. So these kind of things you need to remember. You need to remember the scientist and the crop, the year also. So that will be the history part. And if you come to classification of fungi, many students ask this, that what to cover in the classification. So in case of classification, if you can remember the name of the order up to order, it is enough for the examination. You need not to go for families or suborders or genus, species. That will not be very much relevant. So if you can remember up to orders, under which kingdom, under which order is coming, that will be much enough. And please try to remember what are the fruiting bodies, what are the spores that each microbes is producing. For example, if you take powdery mildew, which spore it is producing, or which this one fruiting body it is producing. For if you take rust pathogen, what are the spores that you're producing. So that if you can remember, that will be much enough. And go through this certain like epidemic diseases, endemic diseases. Please remember the examples for each one of them. We have sporadic, we have endemic, epidemic. Please remember the examples also. And virus. So regarding virus, if you can remember, please remember, try to remember the transmission of viruses, whether it is systemic, whether it is contact. Okay, please try to remember these things, as well as the genetic material of the virus. There are certain viruses who are having ssDNA, ssRNA. So which virus, only the major viruses you can remember, please remember, because there are lot many viruses and remembering all those will be difficult. But major plant pathogenic viruses, please remember the genetic material also, because we see lot of questions regarding the genetic material of the viruses. So please try to remember that. And regarding this chemicals, many ask that, sir, whether we need to remember all the chemicals. Remembering of all the chemicals is not much of importance, because if you can remember the basic one, like organic, like it may be Bordeaux mixture, so those kind of things, what is the composition of Bordeaux mixture, so that will be asked again. So if you can remember those, please remember that. And also in chemicals, remember those chemicals which are common for certain group of diseases. For example, we have hexaconazole or propiconazole for rust diseases. Okay, so these kind of things, if you can remember, that would be enough. And for sulfur, we use sulfur dust for powdery mildew. So these kind of major diseases and just a common fungicide, if you can remember, that would be much enough. So no need to remember all the chemicals or fungicides, and no need to remember the different classification also. There are different classification of fungicides, so that would be very rarely asked. But if you want to remember, you can. But if it is going too much, you can leave that one also. And there is one more source of pathology, is that if you can find any PDF from Angro, we have Angro University, they release the notes. So if you can find PDF for Angro also, that would be very good for the basic 2 courses. And for the diseases of field crops and horticulture crops, so majorly you need to cover is that what are the different organisms, they will ask the scientific name of the organisms, they will ask, and each disease will have different synonym names. For example, they will have some local names, they will have some international names. A single disease will have 3 to 4 names. You must know all the names, and you must know the scientific name of the pathogen. and you must know what is the fruiting body or what is the spores that the pathogen is producing. So that will be important, and please go through the rust. Rust is very much important for examination point of view, especially the wheat rust. There are different types of wheat rust, so please go through wheat rust also. And how the ascus will be developed, how basidium will be developed, what are the spread, how they are spreading, that is the mode of dispersal we have. So go through the mode of dispersal, mode of survival. They mainly ask the examples for mode of survival and mode of dispersal. So if you can go through this, that will be enough for plant pathology. And if you want an objective book for plant pathology, so please go through Utpal Kumar Bhattacharya. Utpal Kumar Bhattacharya is a very good book. So please go through that book for objectives in regard to plant pathology. Now coming to seed technology. So seed technology, again it is almost related to the plant breeding techniques. And seed technology, there is a standard books, that is PK Agarwal. We have PK Agarwal seed technology, and there is a book by Ranganayaki, which is the major objective books for JRF preparation. Objectives of seed technology by Ranganayaki. And there is one more book by Venudevan, and there are other 7 authors. So if you can find these books for seed technology, so that would be more than enough for seed technology. And if you have your class notes also, please refer your class notes, because in seed technology it is very minimal subject. It is not of too much of vast, so it is a very minimal topics are covered in seed technology. So please go through all the topics, read line by line. So in seed technology, line to line reading is required, because there are very precise questions that they are asking in the examination. So go through the years, it may be see when the seed development committee has been developed, when the seed control order has been came, who was the father of seed technology, it may be international level, Indian level. Please go through things, and isolation distance of different crops. For example, if you take maize, there are different isolation, like 400, it may be 200, it may be. If you have different kernel color of maize, again the isolation distance differs. Okay, for what is the isolation distance for self-pollinated crop, cross-pollinated crop, what is seed multiplication ratio, what is seed replacement ratio, what is seed replacement ratio for hybrids, what is for varieties. So please go through these kind of topics. And tags, again the seed tags, we have white tag, foundation seeds, we have certified seeds, and different tags for each class of seeds. So please go through the color of each tags for different class of seeds, because repeatedly we are going to get the questions from the seed tags. So please go through the seed tags. And regarding the seed processing machineries, so please go through the principles of each machinery. For example, if you have a magnetic separator, what is the principle behind the magnetic separator? We have a separation based on texture, separation based on shape, separation based on the surface characters. So these are the different principles that are applied in seed processing machineries. So please go through these aspects. So seed technology, again you need to refer line to line. Please go through seed technology book also. So these 3 books will be enough for seed technology. And coming to microbiology, again microbiology will be little bit overlapping with pathology, because you will be reading the same fungal classification, it may be bacterial classification in microbiology book also, as well as pathology also. So it won't be a much of difficulty for go through microbiology. For microbiology, I would suggest a standard book, that is Taro and Kapoor. Taro and Kapoor is a very standard book for microbiology. Go through that, and if you can get a PDF from Angrum, so go through that also. So that would be, those 2 books will be major books for microbiology. In microbiology, what you need to cover is again the history of microbiology. There are different, like spontaneous generation theory, we have the disapproval of spontaneous generation theory. So these kind of things regarding the scientists, who are the scientists that they were discovered. So those kind of things you need to cover. Again, there are nutrient cycles like N cycle, S cycle, C cycle, phosphorus cycle. So please go through the cycles. And nitrogen metabolism plays a very important role for this examination, because there are many questions that have been asked for nitrogen fixation. It may be biological nitrogen fixation, chemical nitrogen fixation, rhizobium. Different kinds of rhizobium are there. Azorhizobium, bradyrhizobium. We have different types of rhizobium. So please go through rhizobium also, and please try to remember examples for each of the rhizobiums. So you will get precise information in the textbook, whichever the Taro and Kapoor. So please go through Taro and Kapoor completely. And industrial microbiology, again, in industrial microbiology, suppose we have a beer preparation, suppose we have a cheese preparation, mushroom preparation. So no need to remember all the procedure, but what you need to remember from that part is, you need to remember what are the microbes that they are using. That is, the scientific name of the microbes is very much important, and whether there is any specific temperature they are using or specific conditions that they are using. So So those kind of things will be very much important. Go through mushroom cultivation very precisely. So mushroom cultivation also we see a lot of questions like which are the poisonous mushrooms, which are the edible mushrooms. So these kind of questions will be repeatedly coming in the examination. So please go through the mushroom cultivation very precisely from microbiology. So these books will be enough for microbiology. So yes sir, that will be for plant pathology, microbiology.
One part of that examination is also general agriculture.
Yes.
So what is the strategy to cover the general agriculture?
So general agriculture, general agriculture is a common for all the
For all
students.
subjects.
Okay. And many of the students, what they do is they neglect general agriculture. So that is the biggest mistake the students do. As of me, the biggest mistake any student could do is neglecting general agriculture. Because general agriculture, if any student is good in the basic concepts, obviously he will do good. But whoever is doing good in the general agriculture and whoever is having a little bit of disadvantage in the other subject, they could cover up everything in general agriculture, because in general agriculture all the subject you need to read. So in general agriculture, I would prefer major books like Nemraj Sunda. Please read line to line. If you can repeat it as many as times, please repeat the Nemraj Sunda. And Arun Katyan. Arun Katyan is very good book for plant science, especially for plant science. Go through Arun Katyan and don't leave whatever, like if you have read genetics from BD Singh, don't leave genetics from Arun Katyan. Please read, you will get extra information. And we see some questions which have been directly asked from the Arun Katyan genetics. So please don't leave those concepts, read complete book. And for these two will be mainly for general agriculture book. And Nitish Ranjan, there is one more by the plant science by Nitish Ranjan. So if you can read these three books will be enough for general agriculture, completing the general agriculture. Then for objectives regarding the whole topics or whole subjects, you can prefer Ajit Kumar Singh. Ajit Kumar Singh is a very good book for objective kinds. And S.R. Kantwa, please don't leave S.R. Kantwa. S.R. Kantwa is Bible for objectives. S.R. Kantwa is a very good book, and at the last they have given some model papers also. So please solve the model papers, and this will be enough. And one more thing that you need to keep in mind is you also should cover the current affairs, little bit of current affairs which are being just 2 or 3 months prior to examination. You need to cover the current affairs. You can take current affairs by Roop Singh Maitri. There is a book by Roop Singh Maitri, and there is one more current affairs book by Vishal Publications. So you can purchase that. There you can find whatever the statistical data we have. For example, the production technologies, what is the productivity of India, what is the production, which state is having, which crop is having highest export, import, what is the GDP through agriculture. Okay, so these kind of things you will get through current agriculture book. Please go through these topics also. So you will be able to see questions from these kind of things also. And this time what we faced is we found many of the common sense kind of exam questions. For example, the gir forest is for which animal? So that is a simple common understanding question. And what tree is used for note making or paper making? So these kind of things usually you won't get it in the books. So you need to follow some, like you through Instagram, through Facebook also you find some users, like it may be we have AgriScience, we have ICR, JRF. There are some groups. So if you can follow those groups, if you can keep on updating with those kind of things, you will get to know bit information. So please follow those things also.
So one last question, Jayan. What is the appropriate time to start preparation for plant sciences? Since plant sciences has all 5 subjects, and all 5 subjects are major subjects, their syllabus is huge, and the book and the strategy you told, I think it needs proper time and revisions. So what is the appropriate time to start?
So as I said until now, that it is a very vast subject. That is, it is having a 5 major subjects, that to 5 major subjects, and you need to give an appropriate time for each subject. So I think as I prepared is when I started the 4th year, when the 4th year started, when I came to the 4th year, I started my preparation since 4th year. So it took me nearly 8-10 months, you can say. So but in the middle we had some of the programs, like maybe Rave programs we have some extension programs we have. So with regard to classes and all, we need to manage both the things. So it would take around 8 months. So 8 months will be required for proper preparation.
So full 1 year students because they have to revise also, they have to make notes also.
Yeah.
So those who are watching us, if you have any doubts regarding the preparation strategy, though Jayant has elaborated it in a very nice and wonderful manner, each and every strategy he has quoted, but if you have still doubts, you need clarity, so please comment on our comment box.
Yes, sure.
So we are in regular touch with Jayant. We will make one new episode for you if you have a certain valid confusions and doubts. So all the best for you, and you should start your preparation after completing third year of your graduation. 1 year is sufficient. Be in touch with us and secure good ranks, and we will be very happy if we see some of you who are watching our videos as IRA students. Thank you so much.
Thank you.
Namaskar.